So, all right, so here we are talking about heredity. This is just a review for 11.1, 11.2. I'll add some things to kind of help you out as well. So, as we mentioned several times in class, genetic information is passed from parent to offspring, and the determining of a trait is determined by probability. Probability is the number of possible outcomes over the desired outcomes. And so if you think of a coin flip, you want heads. There's one possible outcome out of two that are available to us, heads and tails. Each parent has DNA that comes from the chromosome and it goes to the offspring. And it's this process of sexual reproduction that actually does that. If we do it through asexual reproduction, we're just making a clone. It's a process of mitosis, so it's not going to be very different. All right, so what we're going to look at is genetics, the study of heredity. Heredity is how genes are passed on. In order to do that, we're going to look at the godfather of modern genetics, and that's a priest by the name of Gregor Mendel. And so I think Mendel's story is a pretty interesting one for those who may not have read about him. He was a monk in Czechoslovakia, and he was the gardener. Now, when we think of gardener, you think of like a small patch. He actually maintained the crops for the entire monastery that they would eat and consume. So really, he was a farmer. And his skill comes from, or the reason why he was very good is obviously he's intelligent, but also his life experience. Mendel was an individual that his life experiences kind of added up to basically allow him to be the famous person that he is today. He did some experiments, and from those experiments, he came up with some simple rules or predictions to be able to predict heredity. And we still use those predictions, Mendelian predictions, today. We call this area of genetics Mendelian genetics. He published his work in 1866, but it wasn't found until the 1900s. The reason is he published in he was part of a science club type thing where they would meet. It was like a men's club where they'd meet and do research. It was kind of like a hobby, and he published in that, which obviously was a small journal no one really read. It wasn't until the 1900 that they actually discovered it, and scientists then worked to try to figure out why. Why were those predictions or why were those patterns occurring in genetics? He just identified the patterns, didn't explain why the patterns occurred. So there's Mendel working with the most famous of his species that he studied, which was the pea plant. We'll talk about why it was intelligent for him to pick the pea plant and why he did that. So a little more background on Mendel. Mendel actually started his work, and he actually copied another individual, a farmer by the name of T.A. Knight. T.A. Knight was back in the 1790s, but remember, because he was a farmer, he was working more to try to help grow better crops than actually Mendel trying to figure out some scientific research. Okay. So once again, when scientists found out about Mendel's work, they really worked to try to explain and determine what was actually going on. It's through that work of trying to decipher Mendel's predictions that we learned about genes and chromosomes and eventually about DNA and the structure of DNA all the way up to 1950s. So Mendel's work is what really stimulates that knowledge. So Mendel's story, where did he come from? He was a peasant, and because of that, he knew a lot about agriculture and farming. That knowledge is going to help him make some good decisions about his research that he's going to do later in life. Mendel wanted to originally be a teacher, and we're talking about teacher, we're not talking like me, a high school teacher, we're talking more on the college level, more like a professor. Back then there was an exam you needed to take in order to be a professor. He failed the exam, and so he decided instead to study math and science in Vienna. The reason he entered the priesthood, like many people, if you're in the peasant class, you really can't move up, where being a priest, you can live in a group, have kind of be more than just a peasant. And so that was an opportunity that he liked and therefore went into after studying math and science in the University of Vienna. One of the things that was a new concept at the time was the idea that math could be used along with science. Up until this time period, math and science were kind of different fields that didn't have a lot of interacting or synergy between the two. And so most of the ideas were qualitative observations, and the use of quantitative observations was kind of a new phenomenon. So when Mendel returned to the monastery after studying math and science, he decided to apply this idea of quantitative studies, mathematics, into his scientific research at his local science club, as I had mentioned. So what he did is he took Knight's research and he copied it. He did the exact same thing Knight did a bunch of genetic crosses. But instead of just Knight looking for generalizations, he actually took a mathematical approach to it. And he counted the number of offspring, and by doing that, he was able to see ratios or patterns that developed in the data. Mendel chose pea plants. Part of the reason he was able to make a good decision about choosing pea plants was because of his experience in agriculture based on his peasant lifestyle. So there are 5 main reasons that pea plants were a successful species to pick and why Mendel picked them. Number 1, they're easily grown. They're very hardy, and therefore, because they're hardy, it makes more sense to kind of select them as a species. They produce a large number of offspring. This is important to produce a large number of offspring because it really allows for, when you produce a large number of offspring, what that's going to do is give us better sample size. So it's just like flipping a coin. This is what we'll do in lab next time. If I flipped a coin 4 times, I might get 75% of the time heads. Well, does that mean that heads is more likely to occur? No, it's just due to a small sample size. What would be more likely to get a ratio closer to 50-50 if I did it 1,000 times? And so that's where the numbers really come in. When we look at the election, it's the same concept. How close is the election? Well, if we took a small sample of 10 people in the United States, it might give us very different results than we took the overall population. Number 3, normally they are self-fertilizing. And so what this allows for is that we can control the fertilization process. Mendel could choose which plants he wanted to breed with which plants. He could have a plant with a purple flower breed with a plant with a white flower. In other words, when I say normal self-fertilizing, he can control the experiment. They are produced sexually, which gives us variation. And so there are differences that we can physically see. And that's important, when we talk about this, that there's different phenotypes that we could actually identify. Tall versus short, dwarf, what we call the dwarf versus tall, white flower versus purple, yellow seed versus green seed. Those are physical characteristics that manifest themselves because of meiosis. And a short generation time. In other words, how long do they go from a seed to producing their own seed? If we studied elephants that have a generation time of 18 years, in 100 years, you might have 4 generations. For pea plants, they have a generation time about a couple months. You could have a lot more generations, obviously. You could have up to 400 generations in that time period, producing a lot more offspring, a lot better numbers, and you could really see how the species changed where 400 generations versus 4. So short generation time was also a huge benefit. And here's the pictures of the pea plant we typically think of. They do have flowers. And Mendel, what he noticed was that there's a bunch of characteristics that were either or. Now pea plants have other ones that are more of a variety, but he was really paying attention to things that are either or, easy to see. The plant was either tall or was dwarf. The plant either had what we call it inflated or constricted, green versus yellow, axial versus terminal, purple versus white, all these different things. And so there wasn't an intermediate. It was black or white. It was, up or down, whatever you want to, left or right, 50-50 chance of those two things. That's not necessarily what the probability would be, but that's what he was focusing in on. The fact that he focused in on these things and the fact that he picked pea plants, those are the huge benefit and why he was able to find out his ratios. If he would have picked out characteristics that were not either or, he would not have seen those ratios. His knowledge based on his peasant lifestyle was what allowed him to realize that these are what he wants to focus in on. And here we can see this. Here, obviously, a little bit of a typo. Those should be white up there, but you can see what I refer to. Everyone can kind of tell when I tell purple versus white. The ones that are a little bit different axial, notice it comes out, the flower comes out of the middle, terminal, it's at the end. Green versus yellow, This is a wrinkled versus round, inflated versus constricted, green, yellow, and so on. The dominant characteristics are found here. The recessive characteristics are found there. Okay, so we have to have symbols to represent this. When we talk about this, your P1 generation is the parent generation. Your F1 is the first generation across of the parents. And then finally, the F2 is a cross between two individuals from the first phyla. When he did this, what he did is he made sure that the homozygote, he would take a homozygote, what he called actually a true breed. And the true breed means it's homozygote for a particular characteristic. He took a purple homozygote and crossed it with a white homozygote for flower color. And that would give him the F1. He would then take two offspring there, and that would give him his F2. So, and this is just basically following his procedure. You can kind of read along with this, make sure it makes sense. By true breeding it just meant a purple flower mated with a purple flower, you got all purple. If you were to do this and any of the different characteristic would appear, he'd throw out those plants and would start again. So in other words, if he crossed a white with a white and they produce even one purple flower, he'd get rid of those. And so he'd do that for generation after generation, and that's what produced his P1 or his parental types that were true breed for those characteristics. The key to this down here, this idea that the P generation only displays one particular characteristic or trait. He would then cross-pollinate two varieties from the P generation, okay? So he would take the example, a green seed and cross it with a yellow seed, though each one being true breed. He'd cross a purple flower with a white. The next generation he would get would be called the F1 generation. Now, when he did that, so let's say we cross a true breed purple with a white, he would get all purple, because purple's dominant. Those are big P, little p. And then what he would do is he'd cross two of these. And then that cross, big P, little p, like that, would be his F2 generation. And it's at this levels where he counted. He counted these F2s. Remember, that's the one that really matters, and it's there's where he saw the ratios. The F1s will always have all the same all the dominant phenotype and the heterozygote genotype. So it's the F2 generation that really matters. Okay, and what he noticed was this. When he did the cross, the F1 generation, one of the traits had disappeared. The trait that did not disappear, he called the dominant trait. The one that did disappear, he called the recessive. He came up with the idea of the law of dominance, saying that when you have one dominant trait and one in our case, we call them alleles. One dominant allele with one recessive, the dominant always appears. That is a simple Mendelian example. So if I cross a purple being dominant to white, which is recessive, and it gets a big P and a little p, the dominant trait will only be the one that appears, and you won't even know that you had the one. He then allowed the F1 to pollinate, and when he did that, what he got was a ratio. He noticed that the purple being dominant, 705, but white reappeared. Remember, the white was completely missing from the F1. It had reappeared. Now, T.A. Knight had done that, and that's not that important. T.A. Knight saw that white reappeared. What Mendel did that was different was he counted. And when he counted, he saw that there was an approximate ratio of 3-to-1. For every 3 purple, there was 1 white. And he kept doing it over and over with different traits, and he kept seeing this 3-to-1. Something that must be up. And you can see it, the numbers here with this chart, 3-to-1, 3-to-1, 3-to-1. It's not exactly 3-to-1. Remember, the reason is when you talk about probability, it's the likelihood of event to occur. The larger the sample size, the better the ratio. Take a look at this chart. What has the closest to 3-to-1? Well, I'd say it's this one right here. And the next closest is this one. What do you notice about those two? Well, they're the highest numbers, closest to 3-to-1. And so it's still a probability. What's the odds that you flip heads? 50%. Well, if I flip it, I could be off. If I flip it 10 times, I have 6. Throws my numbers off. But he kept seeing this 3-to-1 ratio, so where's that coming from? It's a 3-to-1 ratio, but it's really disguised as a 1 to 2-to-1 ratio. The reason is you have one of the 3 one of them, that is a purple, completely dominant, what we call big P, big P. You have two that are heterozygote for the condition, big P, little p, and then you have one that's completely recessive, right there. And so it's only the dominant traits we're seeing in the P1 generation reappearing. There's your 3 purple, 1 white. Remember, what the phenotypic and genotypic ratios can be the same, but they do not necessarily have to. So there's the ratios that Mendel saw. All of this can be explained in detail in section 11.1 in our textbook. And I'm actually looking at the hard copy textbook. It's page 311.